And as Leila have mentioned, I'll be talking about the work that I have done as a postdoc at the group of Christina Paulino at the time when she was still at the University of Groningen. And I will be talking about a very peculiar sodium proton exchanger. The protein in question is SLC9C1, and it belongs to this superfamily SLC9 of sodium proton exchangers. They are important for regulation of various cellular processes, such as adjusting intracellular pH, sodium homeostasis, regulating cell volume, to name a few. The interesting thing about SLC9C1 in particular is that it is expressed specifically in sperm. So this protein was first characterized not that long ago, actually in 2003. And the authors of this study first analyzed the total RNA from various murine tissues using the probe specific against SLC9C1. And you can appreciate that the signal could only be detected in the tissue from testes. Next, they also have raised the antibody against this protein, and they have analyzed proteins found in testes, but also only in sperm. And what they could see is that the signal from the sperm basically is just as strong as from the testis itself, leading them to conclude that the protein probably expresses in sperm. They have also labeled the mouse sperm with this antibody. So on the panel on the left, you can see they have incubated the sperm with the antibody directly, and on the right, they have first pre-incubated the antibody with the competing immunogen. And while on the left, the antibody recognizes both the flagella and the heads of the sperm, on the left, the signal from the flagella is gone, indicating that the signal from the heads is probably unspecific because it was not blocked by the immunogen. And so the conclusion was that SLC9C1 localizes specifically to the sperm flagella. They were also quite interested in what is the physiological role of this protein, and they have created the knockout mice. These mice, they did not show any abnormalities in their development or health. The only issue was that the disruption of this gene caused a complete infertility in male mice. So they produced virtually 0 pregnancies in the female mice. And upon closer inspection of the sperm from these knockout animals, the authors did note that in complete knockouts, the sperm motility was virtually abolished compared to the wild-type animals or the heterozygous knockouts. And also, quite interestingly, a recent study reports the case of the human patient with a family history of infertility and decreased sperm motility. So these are the electron micrographs of the control sperm from the healthy individual in comparison to this patient. And you can appreciate that in case of the patient, the sperm flagella shows this really incredible abnormalities in the flagellar structure. And when the authors tried to identify what is the underlying genetic mutation in this patient, they could identify the mutation in actually SLC9C1. And this mutation causes the exon skipping in the gene, and then results in a 33 amino acid deletion at the C terminus of the protein. And so this goes to say that SLC9C1 is essential for sperm motility and fertility, both in mice and humans. And this is where things are starting to get interesting for us. Unlike other sodium proton exchangers, SLC9C1 has auxiliary domains. And this is something that was pointed out in this very first study that characterized this protein. The authors could identify that the gene for SLC9C1 also encodes, in addition to this transport domain, voltage sensing domain, or VSD, and a cyclic nucleotide binding domain, or CNBD. So in other words, instead of a usual just a transport domain as in other sodium proton exchangers, we would have something like this. And just to point out how unique this is, SLC9C proteins, and there are 2 known homologs, SLC9C1 and 2, are the only proteins known to date which combine these 3 domains. So transport domain, VSD, and CNBD. And again, just to emphasize how special this is, I would like to talk a little bit about the mechanisms of membrane transport, considering secondary active transporters such as usual SLC9s and proteins that contain VSD and CNBD. So in secondary active transporters such as SLC9s, they function according to what is called the alternating access mechanism. And so in this scheme, the transport cycle would start with what is called the inward-facing conformation, where the binding site is exposed to the cytoplasm. And so the substrate can bind to the binding site, which leads to the conformational transition to the occluded conformation, where the binding site is not exposed to either side of the membranes, and eventually to the outward-facing conformation. So the substrate can leave the binding site, and the counterion can bind. And basically, the transporter goes back to the inward-facing conformation, releasing the counterion, and the cycle can start again. And so the defining properties of this transport is that this is active transport against the gradient. So in this example, we have protons pumped against the gradient using the sodium gradient to power this transport as a counterion. Secondly, the protons need to continuously cycle from the inward-facing to the outward-facing conformation in order to fulfill the transport function. And it is generally thought that secondary active transporters are gated by the substrates themselves, so as long as the concentrations are suitable, the transporter would fulfill the transport function. And on the other hand, in the voltage-gated ion channels, such as HCN, the protein would be activated upon membrane hyperpolarization, which would cause the movement of the voltage sensor. This is coupled to the pore opening. And once the pore is open, the sodium ions can flow along the electrochemical gradient. And this transport can also be modulated by cyclic nucleotides. And so in contrast to the secondary active transporters, this is passive transport along the electrochemical gradient. The channel does not need to continuously cycle through different conformations to fulfill the transport function. So as long as the pore is open, the ions can be transported. And voltage-gated proteins are activated by changes in membrane potential. And of course, the question is, fine, this protein has the auxiliary domains, but does it actually function differently? Do these domains give it different properties compared to other secondary active transporters? And in 2018, there was this very beautiful study published, which was probably the starting point of our interest in this protein. And the study characterized the SLC9C1 homologue found in sea urchins, Strongylocentrotus purpuratus, hence SP SLC9C1. And the functional properties of this protein were assayed by voltage clamp fluorimetry. So this is the experimental setup. The recordings were performed in the whole cell configuration. This would be the sodium concentration and pH inside and outside of the cell. And the cells were also loaded by the fluorescent indicator BCECF. So during the recordings, cells were first held at -40, then the voltage was stepped to -100 and back to -40. So what we can see is that once the membrane is hyperpolarized, we can observe the increase in this F480 to F440 ratio, which indicates that the cell is getting alkalized and the protons are being transported out. And then once the voltage is stepped back to -40, the signal is slowly returning to baseline. So this really indicates that the protein only transports sodium and protons when the membrane is hyperpolarized. So voltage sensing domain is functional and it does modulate the protein activity. And next, the authors of the study performed this measurement over a range of voltages in presence of cyclic AMP and cyclic GMP. And what they could see that while in the absence of cyclic nucleotides, the protein activates with a V-half of about -70. Cyclic AMP, but not cyclic GMP, shift the V-half closer to the resting potential, such that it's about -56. And so this concludes that CNBD is also functional and it does modulate the transport activity. And we were, of course, very interested, how can a secondary active transporter, such as SLC9C1, be activated very much like ion channels and gated by voltage and cyclic nucleotides? And we thought to solve this question by solving the structures of this protein. We have started with screening a number of homologs for their expression and stability upon extraction. So this is just a sequence alignment where I have marked different phylogenetic groups. And with these thumbnails, I'm indicating the homologs that we have tested for their expression. So you can see that we've mostly focused on mammalian genes, but we have also included this sea urchin homolog, which was characterized in the 2018 study. And fortunately or not, mammalian homologs did not really express in our hands. And the sea urchin protein was the only one that we could express in amounts sufficient for structural work. And here I would like to mention that our group was not the only one that was interested in these proteins, and currently there's 4 papers reporting on the structures of the sea urchin SLC9C1. And if this topic interests you, I do encourage you to check out the other stories as well, because every story contributes a small piece and a different perspective to this problem. But this is the general architecture of SLC9C1 as seen in our structure. The protein assembles as a dimer. So on this figure, you can see one protomer colored by domains, and the second one is shown in gray. And this is quite typical for sodium proton exchangers. They are dimers, so we were not really surprised there. We could also identify all of the functional units that I was talking about before, namely the transport domain, or TD, shown in light blue here, then VSD in dark blue, CNBD in light purple, and also some of the domains which are not usually found neither in SLC9 transporters nor in voltage-gated ion channels. One of them is the additional beta role, which is found C-terminally from CNBD. So we termed it beta CTD. And also quite an extensive network of cytoplasmic helices shown in yellow. We also refer to them as coupling helices, or CH. And I'll walk you through all of these domains one by one, starting with the heart of the transporter, the transport domain. And here the fold is quite typical for other secondary active transporters and sodium proton antiporters. If we look at the protein from the top, we can see that individual protomers within the transport domain, they can be split into 2 subdomains. They're termed dimer domain here in green and core domain in blue. And the function of the dimer domain is thought to be the dimerization of the protein within the membrane, but also providing this rigid scaffold along which the core domain would be able to move up and down in the membrane during transport. And the core domain contains the ion binding site, so it is located on this unwound stretches on helices 5 and 12. And if we look at the surface representation of the core domain, we can also see that there is the cavity extending from the binding site into the cytoplasm, indicating that the transporter is in the inward-facing conformation. And if we take a closer look at this binding site, so there are residues that are known to be important for binding of the substrates, so the threonine, aspartate, another aspartate, and asparagine, and particularly this asparagine and aspartate, ND. They're considered to be a signature motif of electroneutral sodium proton exchangers. Then if we look at VSD, on the first glance, it looks like a usual VSD, and I'm sure a lot of people in the audience are familiar with this fold. It's quite typical, so it consists of 4 helices, termed S1 to S4, and S4 is the voltage sensor, which carries these numerous conserved arginines, which sense the changes in membrane potential. If we look at the sequence alignment of SLC9C1 with other voltage-gated channels, we can appreciate that the number of gating charges almost matches that of HCN1 and HCN4. And like other VSDs, we also have the gating charge transfer center, GCTC, with two conserved countercharges and this aromatic residue here. In our case, it's a tyrosine, which forms the lead on the GCTC. And this helps to coordinate the gating charges as the voltage sensor moves up and down in the membrane. But if we look at the VSD in the context of the entire protein, there are quite marked differences between ion channels and SLC9C1. So this would be the structure of HCN1, and I'm just showing two protomers for clarity. This helix would be S4, and we can see that S4 is really directly coupled to the pore domain, and in some way it is quite intuitive to explain how S4 movement would cause the opening of the pore in case of HCN1. If we look at the protein from the top, we can also appreciate that VSD is quite tightly packed against the pore domain, so there is this direct proximity of the two domains. In case of SLC9C1, so this is the S4 helix. So first, quite remarkable feature for us that we notice is that this helix actually has a very long cytoplasmic extension. It's almost 60 angstrom length in length, just the cytoplasmic part, which was not seen in other structures before. And if we look at the protein from the top, we can also notice that there is a substantial distance between the VSD and the transport domain. So something about 15 angstrom. And actually, there is virtually no interaction between any of the residues of VSD with any of the residues of the transport domain. So we were quite puzzled, how can voltage sensor movement activate this transporter? Now, since our structures were solved in lipid nanodiscs, we were also interested whether we could identify some interactions between protein and lipid, especially given this huge gap between the transport domain and the VSD. And so this is the map of our protein in lipid nanodiscs, where protein is displayed in blue and the nanodisk density in yellow. So if we slice through the extracellular layer and the intracellular bilayer, we can see that there are really concentric layers of lipids in both membrane leaflets, perhaps more pronounced on the extracellular leaflet. What we could also notice, if we look at this region between the VSD and the transport domain, is that the lipids seem to be somewhat better resolved, and we can actually identify individual acyl chains as opposed to this continuous density of lipids elsewhere in the map. In addition to that, we could identify two lipids bound within the transport domain on the extracellular side of the membrane. And this appears to be a common feature in SLC9 structures and also other secondary active transporters. So more often than not, there are lipid molecules bound. But we do not know the identity of these lipids. We have also solved the structures of the protein in detergent. And actually, quite surprisingly, in contrast to nanodisk, where we could only observe one state, we could identify two classes in our detergent dataset, which returned symmetric and asymmetric. And symmetric class looks somewhat rhomboidal from the top, and asymmetric sort of triangular. Actually, this difference is caused by the difference in conformation of S4. And if we look at the model from the side, so what we saw is that in one of the protomers in this asymmetric class, S4 kinks towards the transport domain and really comes into direct contact with this unit, whereas the rest of the domain is not visible in our maps. So we do not think that this conformation is in any way physiologically relevant or reflects any regulatory mechanism, but it rather reflects perhaps the high mobility of the VSDs within the membrane and flexibility of this region. And we do think that detergent environment is in some way softer than the lipid bilayer, and it is not rigid enough to stabilize the VSDs in the upright conformation, as opposed to lipid nanodiscs. Next, talking about the final functional unit, CNBD. The fold of CNBD in SLC9C1 is quite similar to other available CNBD structures. So this would be an overlay of our CNBD in light purple onto the mouse HCN2 CNBD in dark purple. And you see that they do overlay quite nicely, except for this region here, helix C. And we do know from the structures of other CNBD-containing proteins with and without nucleotides that helix C is rather mobile in the absence of nucleotides, but then once the nucleotide binds, it really covers the nucleotide binding site, forming a sort of a lid on top of it. And you can appreciate that in case of SLC9C1, the C helix is positioned closer to the nucleotide binding site, even in the absence of nucleotides. Next, this additional beta role domain, beta CTD. As a beta role domain, it does look similar to CNBD, except for this region here that I have highlighted in yellow. So this region is termed the phosphate binding cassette, or PBC, and it is essential for binding of the nucleotides in CNBDs. And we can see that this region is actually absent in the beta CTD, and it's rather replaced by a short loop. So we do not think that this domain can bind nucleotides. But if we look at the whole structure and how these two domains interact, we think there might be some interactions between the two beta CTDs that really stabilize the dimer interface in the cytoplasmic side, and then really interlock the two cytoplasmic domains and prevent much of movement. But this is, of course, just a hypothesis at the moment. And last but not least are the coupling helices. If we look at the structure of SLC9C1 in its entirety, we can appreciate that these coupling helices, they really occupy the central position in the molecule. And also, even if we just look at the primary sequence, you can see that these coupling helices, they intersperse the three functional units of the protein. If we look at them from the top, we can see that these helices, they arrange as pairs of helices, so 1 and 2, 3 and 4, 6 and 7, 8 and 9, and they form this rhomboidal structure right under the transport domain. Because of their central location in this molecule, we think they can have a twofold function. First of all, we think that they might contribute to locking the transport domain in the inward-facing conformation and preventing the transport activity unless the membrane is hyperpolarized. And we think this can be achieved by two types of interaction. So first, if we look at this helix pair 1 and 2, we can see that it is sort of wedged between the helices 8 and 9 and CNBD of the neighboring protomer. And then on the other hand, there could also be interactions within one protomer between CH4 and the core domain of the transporter. So if we look closer at this region, we think that there might be a network of salt bridges that stabilizes this inward-facing conformation. And then, given the central location of the helices, we think that the second function might be kind of sensing and also communicating the S4 movement to the other two functional units. I have mentioned before that cyclic nucleotides modulate the activity of SLC9C1. So if in the absence of nucleotides, the protein is activated with a V-half of about -70. Upon addition of cyclic AMP, the V-half is shifted to -56. And so Martin, a postdoc in Christina's group, he has purified the isolated CNBD domains of SLC9C1, and he performed ITC measurements with cyclic AMP and cyclic GMP. And what he could notice is that cyclic AMP binds with higher affinity, so it's 2.4 micromolar Compared to cyclic GMP, this would be 9.2 or so. Martin has also performed thermostability measurements of the purified domain alone and in presence of cyclic nucleotides. And he could see that both of them do stabilize the protein, but cyclic GMP to a lesser extent than cyclic AMP. We have also solved the structures of SLC9C1 with both cyclic nucleotides, and we could see the density for the nucleotides in both of the maps. And consistently with other published structures of CNBT domains with cyclic nucleotides, in our case as well, cyclic AMP binds in the anti and cyclic GMP in syn configurations. But that being said, with the information that we have now with our structures and measurements from Martin, we really do not know how exactly SPSLC9C1 discriminates between the two nucleotides and why cyclic AMP is a stronger agonist compared to cyclic GMP. But if we look at the effect of the nucleotides on the global conformation, so I'm just showing unsharpened intermediate processing maps of all three data sets, and we can see that cyclic GMP bound map is quite similar to APO, whereas in cyclic AMP, this density for this C-terminal beta CTD is actually gone. And to us, this indicates that this region displays higher mobility. And to better understand this movement in our data, we have performed the 3D variability analysis. And this is basically the analysis that allows you to split the movement within your data set into several components and visualize it in the form of a movie. And I'll show you the movie just for one of the components. So this is what we observed. It's really disengagement of beta CTDs, somewhat of an asymmetric movement. But the cytoplasmic domains do move apart upon cyclic AMP binding. And also to better resolve this conformational transitions upon CAMP binding, we have focused on individual protomers, and we could identify two conformations where the majority of the protein domains were somewhat okay-ish resolved. We term them cyclic AMP protomer 1 and protomer 2. And actually, protomer 1 is quite similar to the APO conformation. However, it has the cyclic AMP bond. And now I will show you the morph between these two conformations that we have identified. So first, we can notice that there is a increased mobility of this coupling helices. And if we look at this from the bottom, first we can appreciate that actually the cytoplasmic tip of S4 comes in direct contact with the C helix of CNBD in the protomer 1. But then if we look at the protomer 2, we do see that the two of them move apart. And we think that perhaps cyclic AMP binding actually facilitates the release of S4 from these interactions that it has in the cytoplasmic domain. And with that, we proposed the activation mechanism as follows. When the membrane is held at resting membrane potential, the transport domain is inhibited by the interactions with the coupling helices. And then once the membrane is hyperpolarized, there is a sequence of events leading to the activation of the protein. So of course, the voltage sensor S4 moves down, which causes the disruption of the interface between the transport domain and the cytoplasmic coupling helices. Then also this helix pair CH1 and 2 is released. And the interface between the beta CTD domains is disrupted. And once the transport domain is disinhibited, it can facilitate the exchange of ions. And then once the cyclic AMP is added, and this is something that we do see in our data, that the addition of the ligand causes the disruption of this beta CTD interface. And we think this really just removes one of the barriers that the protein has to overcome on the way to activation, which is manifested in the shift of the V half closer to the resting membrane potential. And of course, there is still a lot of open questions, and there are some of these questions and some of the thoughts that I would like to share with you at the end of my talk. So let's consider the sperm capacitation cascade in the sea urchin, and capacitation is the process the sperm has to undergo to be able to fertilize the egg. At least in case of the sea urchin eggs release the peptide, which is called the peptides called speract and resact, which are recognized by this membrane-embedded enzyme guanylyl cyclase, which synthesizes cyclic GMP. And cyclic GMP in turn activates the GK channels. Efflux of potassium through these channels causes the hyperpolarization of the sperm membrane, activating SLC9C1. And we do know now what this protein looks like, and we have a hypothesis of how it can be activated. And so once SLC9C1 is activated, its activity causes the alkalization of the sperm, which is important for several molecules. First and foremost, this is essential for the activation of the CUT-spur, which is the primary calcium channel of sperm in various species. And calcium is important for initiating the motility response in sperm. But also changes in pH activate the soluble enzyme, soluble adenylyl cyclase, or SACY, which is the primary producer of cyclic AMP in sperm. And this is also an essential signaling molecule in sperm in various species. And among other things, cyclic AMP, of course, positively modulates SLC9C1. And so these two proteins seem to have at least a positive regulation loop where activity of SLC9C1 potentiates SACY, and the activity of SACY potentiates SLC9C1. And actually, it is suggested, but it is not really conclusively demonstrated yet, whether these proteins interact or not. And if they do interact, it is unknown at what site, and whether this interaction is transient or strong. And this is really one of these open questions that we're quite interested in. Now, I have mentioned CUT-spur already, and so thinking of these three proteins, a very recent study actually looked at all three, and they tried to identify whether they're conserved or not in various metazoan species. And so actually, SLC9C1 is often referred to in the literature as sperm NHE or SNHE, and SACY in the scheme is SAC, but it's basically the same. And so what the authors could identify is that in the majority of metazoan phylogenetic groups, all three proteins are conserved. And in those cases where you see that these boxes are empty, this indicates that it's not really a clear-cut yes or no in these groups. So some of the species, they might have an ortholog, others might not. But the conclusion of the study was that probably SLC9C1 and SACY are universal modulators of the CUT-spur channel in metazoa. But that being said, despite this conservation, something for us to keep in mind is that depending on the fertilization mechanism, depending on the species, sperm actually experience really different environments. For example, in marine invertebrates, they're just released in water, and they recognize the eggs using chemotaxis, while this is very different in during mammalian fertilization. And here I would like to cite a paragraph from this review that really, I think, nicely summarizes this idea that it seems that sperm preferentially adopt unique sperm and species-specific signaling molecules and pathways. And even though some of these components are conserved, not necessarily they function the same. And therefore, signaling in sperm is rather diverse and uniform. And so to explain a bit better what I mean that they not necessarily function the same, again, I would like to come back to the sperm capacitation cascade in sea urchin in comparison to the human. And you can already appreciate from the scheme that there is quite a few more actors involved in the capacitation of human sperm compared to the sea urchin. But I would like to point out two differences. So if in case of a sea urchin, this enzyme SUCY is regulated by pH. It appears that in human sperm is rather regulated by bicarbonate, implying that there has to be some kind of a system for regulating the bicarbonate concentration inside the sperm. Then it is well established in sea urchin that SLC9C1 is the primary protein responsible for regulating intracellular pH, and this does not seem to be the case in the human sperm. It is suggested that intracellular pH might rather be modulated by HV1 channels and other sodium proton exchangers, namely NHA1 and 2. So the big question is, what does the human NHE do, and does it actually work the same? And I have to say that answering this question is quite complicated by the fact that mammalian SLC9C1 homologs do not really express readily in heterologous systems. But let's just consider the sequence conservation from what we know already from all of these domains, starting with the transport domain. And as I've mentioned before, there is this important ND motif. And if we look at the sequence alignment of this various SLC9C1 homologs, so this would be the ND motif, we can see that it is not conserved in mouse or human genes. And also I can tell you in other mammalian genes that we have looked at. And this is a big debate whether mammalian SLC9C1s actually can facilitate sodium proton exchange or whether they have some kind of different transport function. Which one? Currently it's unknown. Now, if we look at the VSD, again, SLC9C1 from sea urchin has quite a few more gating charges compared to the mouse or human homologs. So it is suggested that the voltage activation of these proteins is probably different from the sea urchin. And lastly, the cyclic nucleotide binding domain. So as we saw in our structures, and as is known from a lot of other studies before us, this phosphate binding cassette here contains a very conserved arginine residue, which coordinates the phosphate group of the cyclic nucleotide. And actually, the authors of the study, which first characterized this sea urchin protein functionally, they do introduce a mutation in this position. And what they see is that the modulation by cyclic nucleotides is gone in this protein. So here you can see the protein without and with cyclic nucleotides. With this mutation, it does not have the same shift in we have compared to the wild type protein. And again, if we look at the sequence alignment, this is this arginine positioned here. Again, mouse and human homologs, they do not have the arginine residue. And it is questionable whether they are modulated by cyclic nucleotides or not. So what we have now is the protein that, although is homologous to SLC9C1 from sea urchin, it seems to have substantial differences in the key positions that are important for the activity of the protein. So what does it actually do? And this is this big burning question that we have. It's very obvious that it is important because, of course, mutations in this gene, they cause all kinds of abnormalities, and they really result in the loss of sperm motility. But what is the exact molecular function is currently unclear. And with that, I would like to thank my former lab, most and foremost, the SLC9C1 team. So this is all of us. And of course, first of all, Cristina Paulino for being an extremely supportive mentor and boss. Martin for taking over the project, and he was really instrumental at the very end with doing all of this nice biochemical characterization of the isolated domains. And Jan for all of his help with cryo-EM side, and the rest of the Paulino group for helpful discussions. Of course, the funding bodies that supported this research. And thank you for your attention. If there are some questions, I'd be happy to take them.
Great. Thank you so much, Valeriia. It was a beautiful talk. All right, so for questions,
you have two options. You can write your question in the Q&A box, and I will read them out, and Valeriia will answer. Or you can raise your hand, and then Greg will make you a panelist, and you can ask your question directly. All right, so while we're waiting for questions, I actually had a couple of things that I'm going to take advantage of this time to ask. So when you say these mammalian orthologs do not express, do you mean that you don't get any, biochemically nicely behaved protein, or do you just get no expression whatsoever?
So with human one, we got no expression. Well, and we always express the proteins as a fusion to GFP. So this is how we assess whether it expresses at all. And we actually saw no fluorescence in the cells. The other ones, we did see fluorescence, some of it quite nicely localized to the membrane, but we just could not extract anything. So it seems to be quite sensitive to that.
Yeah. And I don't know if this is something that would make sense to try, but like making chimeras with the sea urchin ortholog to sort of try to dissect the functionality of all these different domains, like the voltage sensing domain and CNDB.
I think definitely. I don't know for sure, but I think Martin tried some of that while I was already gone from the lab, but I do not really know what was the outcome.
All right. Well, we have some questions. All right. So Damien Bell asks, fascinating structure function studies, Dr. Kalienkova. As you know from the SLC 9C1 S4 sequence alignments, HCN already has an extended S4 with a serine break midway down S4. One plausible explanation for this break is a repeat of the more typical KVS4 that may have arisen in a DNA replication of this sequence in evolution. Does the SLC9C1 overextended cytoplasmic S4 tail provide any clues, like a further replication on top of HCN's replication, to how this may have evolved? You may have just answered that with your SAC-Y conservation.
My SAC-Y conservation.
So I think the question is if you can get any clues from this from the sequence alignment from the sequence of the S4 extended S4 tail as to how this extended S4 has arisen.
So actually, we have not looked into that extensively, and I don't remember if we were blasting just that particular sequence or not against anything. If that is of any interest, I can tell you that there are some plant proteins which have a similar structure. They do not have the VSD, but they have this whole cytoplasmic network of helices and CNBDs. But again, we also did not check whether they're quite similar to SLC9C1 or not. This would be something interesting.
So next up, we have an anonymous attendee that says, thank you for a great presentation of a fascinating and strikingly beautiful membrane protein. Have you considered if the effect of a cyclic nucleotides is indirect rather than direct, for instance, via post-translational modification? CAMP does activate protein kinase A after all.
I think CAMP modulates a lot of proteins in the sperm, but in case of SLC9C1, we do see a direct interaction, and I think on this protein, the effect would be direct. I mean, it binds, it induces conformational change, so that was our assumption that it is modulating the activation.
Vincent Postis asks, did you try mammalian system or insect cells for expression, I assume?
We tried mammalian system only.
And then Gufran Siddiqui asks, are the binding sites for CAMP and cGMP the same or similar in nature, like hydrophobic or hydrophilic amino acids involved?
So let's have a look. So I think here we probably, whoops, sorry, let me click through it. We can see that in principle, the majority of the amino acids that bind both nucleotides are the same. There is probably one study I can think of where they really pinpoint which residues allow to the protein to discriminate between the two nucleotides, STHK. But none of the residues identified in STHK are actually conserved in SLC9C1, so we couldn't make any conclusions based on that.
really nice story. I was wondering if in the human protein, the CAMP is eliminated, it still could be functional.
Possible. I mean, it's obviously important for something, this protein, but the issue is that nobody so far managed to measure the function, and we don't really know what it does. But I would think that the CNBDs of the human protein probably do not bind the cyclic nucleotides.
Next question is from Damon Frampton. Thank you for a great talk. Have you considered trying xenopozoocytes as an expression system? I understand that they may not be great for structural studies, but if memory serves, there are some plant ion channels that cannot be expressed in mammalian cells, but express really nicely in oocytes.
So this would be also something quite interesting to try. We'll see what Christina comes up with.
I was wondering if anyone has done any, proteomics studies of this protein to kind of try to see if there are, is there a molecular factor that is missing in the, in the mammalian expression system that is necessary for, kind of the assembly of this protein in the mammalian sort of.
So Benjamin Kaup has looked a lot into that. I think he has proteomic studies on the sea urchin sperm and this recent paper from 2023 on human sperm. And they actually rule out some of the signaling cascade components that were suggested before. But I don't think they have managed to identify any kind of interaction from the SLC9C1 that would explain why we cannot express it.
I actually have, well, since we have time, I actually have one more question, Valeriia. Sorry, I'm really hogging this. But I was wondering, have you ever, while you were working on this project, did you ever try to reconstitute this protein and try doing like a flux assay for functional studies?
Yes, we have tried. I have to say I have not tried extensively. I have tried maybe twice. And then Martin took over this part and he was struggling a lot with actually achieving reproducibility with reconstitution and also liposome leakiness. Because as soon as you hyperpolarize the liposome, it really becomes proton leaky. So we could not really see the activity. But from what I know, he has managed to solve these problems, but he could not measure the activity. And his idea is that probably it needs something else, but what it is, we don't really know. Yeah, it wasn't an easy protein to work with.
Yeah. So do in all of your structures, so you see conformational changes in the cytoplasmic domains and in the voltage sensing domains, but do you see a lot of change in the transport module?
We actually don't see much of a change in the voltage sensing domain. Well, at least it's difficult to say in the VSD, in the membrane embedded part, but we don't expect the voltage sensor movement because we don't really have hyperpolarizing conditions. The transport domain, we do not see the transport activity. We do not see this upward conformation. But what we do see is this kind of a breathing motion between the two protomers. So they just move like this in the membrane. And this was reported for some other sodium proton antiporters before. So we don't really know what's the function of this movement. But that's something we observed both in APO and in the cyclic AMP dataset.
Okay, so that kind of tracks with your, the outcomes of your functional studies, that you don't really see transport even in the presence of.
And actually, it would be really interesting to solve the structures even at resting membrane potential, so a little bit hyperpolarized, right, at -40, because what we have is really 0. So that would be cool to know whether we see a similar overall structure or not.
If not, then we should thank Valeriia for a beautiful talk, and we'll see you all next month.
Thank you again for inviting me. It was a pleasure. Thank you. Yeah.